Welcome back to the Cestode lectures. The next two parasites we will cover belong to the same genus, which is Taenia. We have Taenia saginata and Taenia solium. The common name for Taenia saginata is the beef tapeworm, and the common name for Taenia solium is the pork tapeworm. These life cycles are identical for both of these parasites, except for the intermediate hosts for each one of them. And that's how they're named. So Taenia saginata has a cow for an intermediate host, and Taenia solium has a pig. And that's the only difference here, is which animal serves as the intermediate host. So let's talk through it. Starting right here, we have eggs or gravid proglottids that are passed in feces from humans. And you'll notice this is the diagnostic stage, is seeing eggs or gravid proglottids in feces. So they get passed into the environment. Now, either a cow or a pig would ingest the eggs or gravid proglottids, which contain eggs, in vegetation contaminated by these things. So this would be a case of where there's poor sanitation practices, where there's not good plumbing to remove fecal debris, or there are places that intentionally use human feces as fertilizer. So inside each of these animals, depending on which species we have, the life cycle stage to come out of the egg is called an oncosphere. So it hatches, it penetrates their intestinal wall, circulates in the bloodstream, and gets to their musculature. Inside their tissues, the oncosphere will develop into the next life cycle stage called the Cestodes larva, or plural would be Cestocerci. So you can see the larva here coiled up in the animal's tissues. And you'll notice this is the infective stage. So this is how we get infected with Taenia saginata or Taenia solium. It's by ingesting raw or undercooked infected meat. And it's infected with the Cestocerci larva. That is your infective stage. The larva will then develop in our intestines into the scolex. So the scolex comes out of the larva. And here's what the scollices of each one of our species looks like. And we'll talk about the differences. And of course, via the suckers and also hooks for Taenia solium, it will attach to our intestinal wall and grow very long. All the proglottids will grow, and then eggs or gravid proglottids can be shed in feces. So just talking through it again, we have eggs or gravid proglottids passed in feces. They are ingested by an intermediate host, either the cow or the pig. Inside the intermediate host, oncospheres hatch from the egg, develops into Cestocerci larvae inside those animals. Humans get infected by eating undercooked beef or pork containing that Cestocerci larva. And then the scolex attaches to the mucosa of the intestine and the larva matures into adults. So for diagnosing these two species, the more common ways are to see eggs or proglottids in feces. Remember, it's very rare to see a scolex in feces unless the patient just underwent heavy antiparasitic treatment that would be aimed at unattaching the scolex from the mucosa. But in any case, we'll learn how we can differentiate these two species. So the egg is pretty characteristic. It doesn't look like any other eggs you'll see. It's described as looking like a kiwi, a slice of a kiwi, or even a wagon wheel with these radial striations that are coming out from the center. It is embryonated and it's got 6 hooklets, so there's 3 pairs of hooklets that you may be able to see inside of the egg. Now, The egg is identical for both species. So when you see this egg, you don't know whether it's Taenia saginata or Taenia solium, and we just report it as Taenia species. And this is what they look like. So here's your radial striations. It's all these little lines coming out around the edge. This is a great picture of the hooklets. So you see these lines inside of here. Those are the hooklets that we're referring to. So again, all we report here when we see this is Taenia species. Now, the scollices of these two can be differentiated. Remember, it's rare to see the scollices, but we do need to learn the difference between the two. So Taenia saginata has 4 suckers. Taenia solium also has 4 suckers, but it also has an armed rostellum. Remember, the rostellum is this extra bump on top of the scolex, and then when we say armed, it means it has all these hooks. So that's a really cool picture. You can even see sort of the 4 suckers here. 1, 2, 3, 4. So that's the difference in their scollices. They both have 4 suckers, except Taenia solium has an armed rostellum. We can also tell their gravid proglottids apart. The gravid proglottids of Taenia saginata have a lot of uterine branches. So the uterus in this one it's sort of like the trunk here that's going the whole length of the proglottid. And then you have these lines or these branches coming off of the main trunk. Now, you don't need to memorize 15 to 20 uterine branches on each side of the main uterine trunk. You don't need to memorize that number. What you do need to understand is that Taenia saginata has more uterine branches than Taenia solium. So I know it's hard to count in this picture. Technically, there's about 7 to 13 on each side of the uterine trunk for Taenia solium. But the take-home message here is that if these two pictures were right next to each other, you'd be able to tell that this picture has more branching than this one does. So that's the main point here, is that if you had these side by side, you could tell them apart. Taenia saginata has more than Taenia solium. So on to the genus Hymenolopus. We will talk about Hymenolopus nana and Hymenolopus diminuta. So same genus, 2 different species. Starting with Hymenolopus nana, this is actually the most common cestode infection in the United States, especially in southeastern United States. It is found worldwide. It is common in children and in places where people live in close quarters. Also daycare centers, institutionalized people, hospitalized people where it's very close quarters. Its common name is the dwarf tapeworm. We get Hymenolopus nana we can actually get it a couple ways. We can ingest the egg, like in dirt or contaminated dirt, or insects like fleas that have ingested the egg. This life cycle is unique because no intermediate host is required. There can be an intermediate host, but it's not required. And another reason why it's common in children, or it's easily gets out of control, is that there's an auto-infection branch of the life cycle that can occur as well. So we'll go through these 3 methods of transmission for Hymenolopus nana. In this first one, the eggs are infective. So make a note of that. This is in this first method, the infective stage for humans is the egg. So this would be where we have fecal oral route of contamination with human feces. So a human ingests the infective egg. The egg hatches in human intestines and it forms into a cysticercoid larva in the mucosa of our intestines. So remember in the Taenias, we talked about there was cysticercus larva. When we talk about the Hymenolopus life cycle, it's called a cysticercoid larva. The larva gets into the lumen of the intestines, and out from the larva comes the scolex, and the scolex attaches to the mucosa. The scolex will then grow proglottids and release infective eggs in the feces. The second method of transmission is called the autoinfection or the auto-reinfection. This is when an egg can actually hatch in vivo inside the own person who's infected with the Hymenolopus adult. So you have a Hymenolopus nana adult attached to your intestines. It's shedding eggs, but all of the eggs don't leave through the feces. They actually hatch in the GI tract, and the egg can mature into the cysticercoid larva. So it's continuing maturation within the same host. It never leaves the human host. The third method of transmission is when the cysticercoid larva is at the infective stage. So this is when we have an intermediate host. The parasite may exist in other animals, for example, fleas, beetles, rats, and house mice. These animals can ingest eggs, contaminated infective eggs, and then the cysticercoid larva can develop inside these animals. Then humans get infected by ingesting these animals. And you think, how can you swallow a flea or a beetle? But it can be fairly easy, especially if you have dry foods that aren't well sealed, like cereals or rice or pastas, and you can get fleas in your house and they can set up shop in those dry bags of food, and you could ingest fleas fairly easily and not necessarily know it. So let's talk about what they look like. So Hymenolopus nana, the scolex is armed, has an armed rostellum, just like what? What did we just talk about that also has an armed rostellum? It was Taenia solium. So the next species, Hymenolopus diminuta, does not have an armed rostellum. So that's one way to tell these two species apart. Remember, it's rare to see a scolex, so more commonly we would be diagnosing this by eggs in stool. The eggs are, so to compare to diminuta, which is the next one we'll talk about, the eggs are smaller than diminuta, and they also have polar filaments. And this is what the polar filaments are. So it's got an outer shell and then an embryo inside the shell. And on either side, there's polar plugs. Out of the polar plugs you can see these little strands. These are polar filaments. I know it's hard to see in this one. And you can see the hooks. Remember we talked about hooks for the Taenias as well. So I know it's hard to see size. You'll want to refer to the scale of all the eggs that you have in your note set. But Hymenolopus nana eggs are smaller than diminuta, and they also have polar filaments. whereas Diminuta eggs are larger and they do not have polar filaments. So Diminuta is rarer than Hymenolopus nana. This is truly a zoonotic infection. This almost always is occurring in rats. So this is known as the rat tapeworm. And we're accidental hosts. We are not the normal host for this one. You need an intermediate host for Hymenolopus diminuta, whereas for Hymenolopus nana, an intermediate host was not required. We get Hymenolopus diminuta by again ingesting fleas containing the infective cysticercoid larva. And the fleas got infected by feeding on the feces of rats. So the way to control these is to have good pest control, good rodent control, and then to make sure you have well-sealed food and everything. So that's really the main takeaway here is the infective stage, it's known as the rat tapeworm, and how to distinguish the eggs from Hymenolopus nana. Remember, Hymenolopus diminuta eggs are larger, and they are very similar looking except no polar filaments. And you can see the nice clearing around this inner shell here. Whereas Nana had those polar filaments.